Hello everyone, my name is Cassidy Coker and I am a senior biochemistry major from Abilene, Texas. Today I will be discussing the elemental, isotopic, and morphological measurements as proxies for plant physiology in 50-year-old herbarium specimens. So let's go ahead and delve into this presentation. So the purpose of this experiment is basically to utilize these 50-year-old herbarium specimens that have been found in the E.L. Reed Herbarium at Texas Tech because they serve as a historical data system to observe changes in somatal density, nitrogen availability, and photosynthetic water usage. And so what we are going to do and what we have done is we are using elemental and isotopic analysis, which is basically mass spectroscopy to analyze the amount of carbon and nitrogen that these plants are using. Additionally, we are using morphological features to basically identify how plant plasticity has changed over time. So the way that we are doing this is we have taken the stomatal densities of each of these plants and compared it with elevation as well as the carbon nitrogen ratio. And we will delve into that later within this presentation. But so currently we have sampled a little bit over 200 different specimens for this project. Unfortunately, only about 50 of those are included in this presentation because this is what we have the carbon and nitrogen ratio on. And so of these 45 to 50 specimens that we have in this project, they are going to range over forbs, grasses, shrubs, and trees for their life history. And so let's go ahead and move on to our future objectives for this project. So right now, our research project is currently hoping to complete our elemental, isotopic, and morphological data analysis for over 200 additional herbarium specimens for a total of 250 different samples for this project. And all of these samples will be found within the Guadalupe Mountains National Park, or GUMO for short. And so we will find a couple of these specimens outside of the park, but the majority of them are found within GUMO. And we will be using mass spectrometry and somatal density measurements to again determine the isotopic, the elemental, and the morphological differences between each of these species. So right now we are comparing and contrasting the elevation over 45 different specimens. However, in the future, we do hope to be able to determine that for 250 GUMO samples within the carbon and nitrogen percentages and stomatal density. Additionally, we hope to use this stomatal density to determine plant plasticity variations among different plant families. So currently we have tried to take plants from virtually every single plant family that we have within our GUMO collection. However, only 11 different families are included in this subset of data that we have today in this presentation. And of course, in the future, we do hope to visit the Guadalupe Mountains National Park once again so that we can go ahead and harvest some of those plants that we have already taken over 50 years ago. That way we can compare them over time and see how plant plasticity has changed. So in order to determine the isotopic and elemental analysis of the different herbarium specimens, what we did was we sampled a variety of forbs, grasses, shrubs, and trees so that we had a wide variety of different samples. And so currently for this presentation, we have sampled 45 different herbarium specimens. And what we did was we took a small amount of leaf tissue, and once we had this leaf tissue, we went ahead and determined the area and the mass of these specimens so that they could further be used for isotopic and elemental analysis. Once the area and weight was taken for each of these samples, we went ahead and ground it into a powder, and then this powder was used for further analysis. And so before we could actually go through with mass spectrometry, we had to do something called ball foiling. And so it's basically taking a little bit of tin foil and placing our powdered sample into it, folding it up so that the carbon and the nitrogen is preserved within that sample. And what we do is we place this kind of tin foil ball, we like to call them DNA burritos, and we place it in the mass spectrometry, and that's whenever the machine will analytically analyze the specimen for us, and we will determine the carbon and nitrogen ratio. On the other hand, in order to determine morphological plasticity of the herbarium plants, what we do is we take a little bit of nail polish, clear nail polish, and we take the plant that we have already sampled and we will paint it over the leaf tissue. And once it has dried, we will remove that nail polish and we will place it underneath a microscope. And that way we can count the stomatal density within each of the plants. It does take quite a big of a large amount of magnification to determine that. And then we basically piece together all of the stomatal counts to get a good idea of what the entire stomatal density is for that specimen. As we move on to our results of the experiment, let's go ahead and look at the map. So we did mainly take a lot of our plants from the Guadalupe National Mountains Park because that we have over 20,000 different specimens within the E.L. Reed Herbarium. And so it made sense to take a lot of those from that park. And so we also have about 4 taken from Carlsbad Caverns. And this area right here is our 9K ranch where we have sampled them over 50 years ago. In comparison, we have went ahead and graphed the stomatal density. And so this graph is based on elevation. And so as we can see how the stomatal density has changed based on elevation. This area right here called McKittrick Canyon is going to be one of the higher elevations. And as you can see, there is a higher amount of stomatal density in that area. As we go kind of lower within the park to where most of the plains are, we will see that the stomatal density has decreased in that area. The same for the Carlsbad Caverns. As we go up in elevation, we will see a greater amount of stomatal density in each of our plants. And then of course the 9K ranch is going to be a lower elevation. And like I said earlier, we will see very low stomatal density in that area. Let's go ahead and move on and analyze some of our carbon-nitrogen ratios that we have determined. As we are looking at our different species compared to our carbon-nitrogen ratio, you can see that a lot of our shrubs are found in the higher aspect of the carbon-nitrogen ratio. So, as we've outlined in our conclusion, the higher carbon-nitrogen ratio does indicate less metabolic activity. Because the plant isn't using as much carbon, it is storing it. And that is basically saying that this plant is not using a lot of its metabolic activity and not performing photosynthesis at a rate that another plant might. That might be a part of a different life history. And so we can see that trend for trees as well. And this could be postulated because these plants are bigger and they can store more energy because they have a larger root system. Additionally, whenever we compare it with the grasses, you will see that the grasses do have a lower carbon-nitrogen ratio. And just like the opposite of the shrubs and the trees, the lower carbon-nitrogen ratios do indicate a greater need for photosynthesis and metabolic activity. And our forbs, as we have outlined in these pictures right here, this Asclepia asperella is a forb, and it is going to be a lot smaller than a tree. It is not going to have as great of the root system. So that means that it is going to have a greater metabolic activity, and that is demonstrated within this graph right here. As we compare stomatal density with our percent nitrogen content, you will see that for our grass, shrub, and trees, as our stomatal density increases, our percent nitrogen content also increases. However, for forbs, it does decrease, and we hope to obtain clearer results in the future whenever we have a larger population size of this result. And so using ANOVA statistical analyzation, we will see that if we compare stomatal density with the nitrogen content, as well as life history with the nitrogen content, these two alone, the probability is much greater than the F value. And so we can say that it is not statistically significant between those two. However, if we compare the stomatal density and life history with nitrogen content, we will see that there is a significant interaction between the two. Moving on to where we can compare elevation with the carbon-nitrogen ratio, we will see that there is a trend to where the higher the elevation, the less carbon-nitrogen ratio there is. And that is because plants at a higher elevation are needing to use more energy, and because they are closer to the sun and they are losing that water. However, whenever we move down in elevation, we will see that plants are conserving their carbon and nitrogen more than those who are not. All right, and the references. So we did obtain all of our results from the TORCH database. This is our database where all of our GUMO specimens are located, as well as all of their localities. Additionally, we used ImageJ to determine the area of each of the plants. We also used a couple of papers for kind of an idea of how to use this isotopic and elemental analysis and mass spectrometry so that we can analyze some of these different variables within each of our plants. Lastly, I would like to thank the Honors College Undergraduate Research Scholar Program supported by the C.H. and Helen Jones Foundation, because they have supported each of us throughout this time period. Over the past 2 years, I have gained so much knowledge through the Honors College, and it has been one of the highlights of my collegiate career. Additionally, I would like to thank the Johnson Lab, as well as specifically Dr. Matt Johnson, for his constant encouragement and guidance. So thank you and have a lovely day.